So today is the last lecture before the midterm, and also my last lecture with you. Uh, so can you all see my screen?
Yes, sir.
Good. So we are going to talk today about what we are doing using functional genomics in epigenetics lab at LUMS. And during this lecture, you will learn the concept of um, epigenetic cell memory, uh, chromatin gene regulation by polycomb and trithorax group genes. So what you all know that we all start our life from a single fertilized egg, uh, which is shown here. Then this is the day one. This is 3 days old embryo, 4 days, 7, and 19 to 20 days old embryo. This is the time pretty much where, so first 3 weeks or so, where finally gastrulation is, sorry, where, gastrulation is completed. You have 3 different layers, ectoderm, mesoderm, and endoderm formed. And also from here, then different cell lineages, because these 3 layers, which collectively are called germ layers, not germ cells. These are germ layers. Don't confuse them with germ cells. Are formed, and then from here you have different tissues, cell lineages are formed. Which means already at this stage, when germ layers are formed, and recently Wolf Reich's lab in Babraham, Cambridge, they published that when they isolated cells at this early stage and they did RNA-seq, total RNA-seq analysis, they found 3 populations of cells based on just, it was totally unbiased experiment. They collected single cells and then performed RNA-seq from individual cells. And when they clustered, they laid them on the basis of gene expression pattern. What they found 3 clusters of 3 very distinct clusters of cells. And they were actually referring to ectoderm, mesoderm, and endoderm, which means already at this stage, you have very, very distinct gene expression patterns being established. And then as you move on in development, when different cell types are established, you have further specificity at the level of gene regulation. So when you start your life from single cell, by the time, so this is like 23 weeks. So by the time, stage 23, 56 to 60 days. By the time you are born, you have more than 200 different cell types. More than 200 different cell types. And all these cell types, they originated from single fertilized egg, which means the 46 chromosomes here, each cell type, with the exception of germ cells, they also, each cell type contains 46 chromosomes. These are those 46 chromosomes in a diploid cell. And of course, X and Y can be different based on the gender of the baby. However, when each cell contains same 46 chromosomes, it means each cell contains same set of genes. Genes present on chromosomes are entirely same in all the cell types which are present in a fully formed baby. Now, what I just told you already, distinction between different cells, it starts taking place at the stage of gastrulation. Gene expression patterns are changed, and these cell type specific gene expression patterns are then maintained subsequently. For example, when very early during development, cells are going to be determined, their fate is determined. For example, eye cells, there will be very, very specific cell type specific gene expression, which we refer to eye specific gene expression pattern. And all the progenitors of those cells, few early cells, they will pass on through mitosis the gene expression pattern, which is for eyes. Now the question arises, how these cell type specific gene expression patterns are established and how they are maintained. Okay, so this cartoon tells you started, for example, life here, then you were many cells, many hundred of cells, and they start becoming different. So their fate, so we call this stage cell fate determination. And this happens at the level of gene expression. Now, once cell fates are established, cells divide and they pass on the gene expression pattern. So, for example, this cell after cell fate determination is determined to make future liver. So it will keep dividing mitotically before a fully differentiated liver is formed. So here at cell fate determination stage, cells pretty much maybe they look morphologically same, but biochemically at the level of gene expression, the proteins present there, etc., they are different, though all of them contain same 46 chromosomes. However, genes present on those 46 chromosomes, they are differentially regulated. So here, differential gene expression is being established. Once a cell type specific gene expression pattern is established, then when this cell is going to divide mitotically, each cell in a lineage is going to remember the gene expression pattern through cell cycle. So this maintenance of cell identity due to maintenance of cell type specific gene expression pattern is referred to as cellular memory. We also refer to it as transcriptional cell memory. Okay. And there is a time difference. So cell fate determination all the way up to differentiation. So differentiation means now you have differentiated tissues and organs being formed. Here, morphologically cells will look different as well. They will start performing different function as well. So there's a difference. So this takes place, cell fate determination takes place much earlier during development, and then this is a gradual process. The the cellular memory, the process of cellular memory was discovered or is very well characterized in Drosophila melanogaster. This is the fruit fly, the famous model system we talked about earlier during our course, which was brought into labs by T.H. Morgan. So this cartoon shows you this is the early embryonic stage. This is like few hours old embryo. As you know, flies from egg to adult, it takes 10 days. Now, in the genetics of development, we learned that how development starts, anterior and posterior axes are formed, and then there comes a stage within initial few hours of development where fly body becomes segmented, parasegments, and then you also learned about relationship between parasegments and segments. Now here, if you pay attention, this is cluster of homeotic genes. This is the bithorax complex. This is the chromosome 3, and this is antennapedia complex on the same chromosome. This hash line shows you long distance between these two clusters of genes. Now, these are the master regulators of development, and they are involved in giving specific identity to different cell types. If you pay attention, the labial, proboscipedia, deformed, sex chromes reduced, antennapedia, the genes which are in the antennapedia cluster, they are expressed from anterior in embryo towards posterior. So just like these genes are organized 5 to 3 here, their chromosomal organization somehow is going, from anterior to posterior. It has nothing to do with the morphology, but it's an observation, the way they are organized. So for example, labial is expressed in the head region, deformed, then you see sex chromes reduced. Sex chromes reduced is a gene, so which is involved uh, in formation of male specific comb-like structure. Then you have antennapedia. Bithorax complex starts here. You see in the UBX is expressed here. It's a color coded. ABDA is in this region, and finally ABDB. So the gene expression pattern of these master regulators, this is established very early during development at the stage of segmentation or so. And then these gene expression patterns are maintained throughout development all the way to adulthood. It goes through larval stages, pupal stages, and so on. So this is basically at transcriptional level. These genes are, so labial will be switched on, all the others will be switched off in this region. Here, ABDB is active, is switched on, but these cells, they contain same chromosome, same genes, which means all these genes are present here as well. But all the others are repressed. And we learned about relationship between UBX, ABD, ABDB when we talked in genetics of development course. Okay. It's also shocking to observe that the organization of the genes as well as the genes, they are fully conserved when you go from an invertebrate model, which is the fruit fly, to a mammalian system, which is mouse embryo. So these genes are exactly their chromosomal organization and the way they are expressed from anterior to posterior, head to toe, this is fully conserved. So we call this collinearity. They are collinear. So their organization on chromosome is also conserved, as well as their expression pattern is also conserved. Now we call them homeotic genes in fly because if any one of them, for example, sex chrome reduced, it gets ectopically expressed in the region, for example, this region where normally it is silent, you will get sex chromes produced here as well, which means the thoracic segment 2 is taking up identity of thoracic segment 1. If you have ABDB start expressing in this region, A4 and A5 will take up identity of A6. Or if you will have UBX expressed in T2 where normally wings are present, so wings will become haltier. So the homeotic is driven from homeosis. One body segment takes up identity of another one. So this maintenance function, so once they are established, They are then maintained at the level of their transcription levels are maintained, or cell type specific gene expression pattern of these genes is maintained at the epigenetic level. So this cell memory, we call this, it is maintained at the epigenetic level. This is what I explained you already. You have, you know, this is the ectopic expression of antennapedia. So where is antennapedia? So this is the antennapedia. And antennapedia is normally expressed in the legs. But if antennapedia gets ectopically active in the head region, you see legs are present on the head. If you have another gene that's not present in the bithorax or antennapedia cluster, but it's called ILS gene. In mammals, the homolog is BIO6. So this is the normal. The ILS gene is involved in formation of eyes in wild type. But if you have ectopic expression of ILS on the legs using UAS GAL4 system, so you can use US GAL4 system and you can use ILS enhancer to drive expression of GAL4. And under UAS, you can have ILS gene, wild type ILS gene. When you will cross these flies with this fly, sorry, not ILS. ILS, if you will drive ILS GAL4, it will express ILS in the eyes. You use leg specific. And you can use, for example, antennapedia, GAL4, or whichever, distal-less GAL4, whatever, which are expressed in the eyes. So now an ectopic expression of ILS in the legs is going to form eyes. So this is the master. BIO6 is also a master regulator. And this is antennapedia, ectopic expression of antennapedia. Similarly, if you have mutation in bithorax complex, where normally, so wing is present only on the in the T2, thoracic segment 2. And next to wing is a tiny structure, a bulb-like structure, which is called haltear. So if there's a mutation in the bithorax complex and UBX, you have deletion in UBX, you will get an additional thorax, which is, that's why it's called bithorax complex, and you have complete wing formation. Okay. So this picture you already saw in your developmental genetics lectures. Maternal genes, Bickert and Cordell, or the other maternal genes, they lay down the foundation for anterior, posterior, and dorsal ventral axis formation. As you know, they are transient, they activate gap genes, NIPS and CRUPL are shown here, and then a cascade of gene expression, gap genes to payroll genes, and then segmentation genes. And finally, you have homeotic genes. So these genes, bithorax complex and antennapedia, they, in this cascade of gene expression in early development, they come at the end. And they are the ones I just showed you during early development at the time of self-fate determination. Once they are active, now you cannot go on forever, you know, being transient and passing on job or baton to others. So these selector genes, or genes which are giving identity to different cell types, their gene expression pattern is to be maintained so that generation after generation during mitosis, this is passed on and you have an adult which inherits or which has all the segmented body plan showing the proper gene expression patterns are fully maintained and a normal adult is formed. So once all these are gone, the gene expression pattern of homeotic genes is maintained by two chromatin regulators, which we call polycomb group and trithorax group. Polycomb group is also called silencers, and these are activators, or also called anti-silencers. Now, this is slide from my postdoc mentor Renato Paro, and I think this was made by a former PhD in his lab, which is Tariq, maybe, if I am not wrong. So this explain you whatever we have covered so far, whatever we have learned so far. Let's say this is the time of self-fate determination. So far, gene expression pattern is okay. Now, one of the cell is going to go in such a fate, the other one is this fate. Just, this is a perfect rectangle, this is with rounded. So these are different cell types. Now, these two genes for this fate is they are to be switched off, and this is to be switched on for having this cell fate. Let's say this is cell fate A, this is cell fate B. For B cell fate, these two genes are to be switched off, and this is the cell type specific gene which is going to give identity to this particular cell type. After mitosis, the gene expression patterns are maintained. If they were off, they have to be maintained in the off state because they are not required for this cell fate. And the one which is to be on, it should be inherited through mitosis, through cell division in the on state in all future daughter cells, generation after mitosis. And same is case for this one. Now, how do you maintain these gene expression patterns? This is where, so you can imagine these are homeotic genes. You can imagine these, let's say, UBX gene expression pattern is, this is UBX, for example, and here, this will be UBX. This will be off, and here UBX is on. So in order to maintain these gene expression states, so you have trithorax group and polycomb group. So they bind to chromatin. So trithorax group, as I told you, they are activators or anti-silencers, and trithorax group genes, they bind to chromatin and they maintain these genes in the on state. However, polycomb group, they bind to their targets and they maintain genes in the off state. And this is inherited through mitosis, through cell division, and that's why we call this a memory, cellular memory, because cells, they remember the characteristic or cell type specific gene expression pattern which was in the mother cells. So what are polycomb group and trithorax group genes? So polycomb group genes are basically, they are different, they come in different complexes, multi-subunit protein complexes. For example, two very well characterized complexes are PRC2, PRC1. There are others known as well, like 4 repression complex. Now more and more variants of PRC1, PRC2 are also being discovered. However, two very well characterized complexes are PRC1, PRC2. PRC2 complex contains enhancer of zest. We already talked about enhancer of zest when we talked about position effect variegation screen. And sorry, not position effect variegation screen. When we talked about set domain, and we also talked about X inactivation and earlier uh, examples of imprinting, etc. So enhancer of zest, it's a histone methyl transferase. It catalyzes H3K27 due to the set domain. We talked about all these set domain, etc., when we talked about epigenetics in the earlier lectures of epigenetics. Now, there are other proteins as well, SUSI12, ESC, E55 as well. However, once they methylate H3K27, this methyl mark is recognized by, we already talked about polycomb when we talked about chromodomain in HP1 and chromodomain in polycomb, etc. So chromodomain containing polycomb protein, it recognizes this methyl mark, as well as that another protein in this complex PRC1 is called D ring. D ring catalyzes histone H2A, lysine 118 ubiquitination. These are hallmarks of silencing. So if you characterize chromatin and you find H3K27 trimethylation as well as monoubiquitination and H2A lysine 118, you will say these are polycomb targets. Now, this is, I told you there is a 4 repression complex as well. 4 is a protein which is DNA binding protein. These complexes, they don't contain specific DNA binding protein in them, but this is a DNA binding protein as well, 4. Now, in contrast to polycomb complexes, there is a great variation or diversity in the trithorax group complex. Brahma complex, so this complex is a chromatin remodeling complex, which is shown here. Then you have trithorax, SBF, CBP, etc. This is also called TAC1 complex. So it contains histone acetyltransferase. CBP is a histone acetyltransferase, and trithorax is, we already talked about this, it H3 lysine 4 trimethylation. Okay, and H1 containing complex, it H1 catalyze H3 K36 trimethylation. So this is chromatin remodeler. This is, and showing the TAC1 and H1, they are involved in transcription activation to maintain genes in the on state. So this is a simplified cartoon, and I have borrowed this slide from, again, Renato. So this cartoon is actually showing you this picture. It is simplified form of this picture, that when on my click, all of them disappear. They have, after establishing the expression pattern of homeotic genes, they are disappearing now, and these characteristic gene expression are to be maintained by polycomb. So here you can see this transcription factor is basically depicting the early establishment phase of cell type specific gene expression pattern. So they disappear, and the maintenance function is done by polycomb and trithorax. Polycomb and trithorax in late 80s and early 90s, it was shown that they bind to characteristic DNA sequence, very specific DNA sequence, which is cis-acting, which are cis-acting elements, and they were referred to as polycomb response elements. The DNA sequence here was referred to as polycomb response element, and later on it was discovered that trithorax group complexes, they also bind at the same site, and they regulate gene expression by acting on the promoter. And gene will be based on which of the complex is going to overrule. If trithorax overrules, gene will be maintained in the on state. If polycomb overrules, the gene will be maintained in the off state. Now, there is so far what you have learned by my explanation, that if you have polycomb, gene is off, the red. If you have trithorax, gene is on, the green. However, this is not the case. When you read papers, you find that polycomb and trithorax, they both are co-localizing. They both are binding to the chromatin, and genes are off, genes are on, and these states are reversible as well. So if you modulate trithorax group complex, let's say you knock down trithorax group, this on gene will be turned into off state. Or if you knock down or modulate, deplete polycomb group complex or members, this off gene will be switched on. So what then determines whether gene will be switched on or switched off? So 11 years ago, when I came back to Pakistan, we thought that there must be some cell signaling components. There must be some post-translational modifications of polycomb or some additional components of polycomb trithorax, because what we know about them, that does not really tells us which one will rule. So if there are post-translational modification of polycomb group, they may favor polycomb and then gene is switched off. And if there are post-translational modifications or some cell signaling event, not necessarily post-translational modification of trithorax, but some cell signaling event, so that will switch on gene in the favor of trithorax. In mammals, so this is for flies, but in mammals, you should also know that polycomb trithorax regulated gene, so polycomb enhancer of zest, is shown to go together with DNA methylation as well. Cytosine DNA, 5-methyl cytosine DNA methylation as well, which is shown here. In flies, there's no DNA methylation at this stage. Now, this was the working hypothesis. This is a model. This is showing, some extracellular signals or maybe some intracellular signals. Some cell signaling is taking place, and these events, they tell the cell that polycomb is to overrule and polycomb should maintain gene in the off state. And vice versa, trithorax is favored here, and trithorax, in response to certain cell signaling event, is going to maintain gene in the on state. And these states are, we already know, these epigenetic states are reversible as well. So what it tells us, that what we hypothesize, that there are some additional components. There are some maybe not core components of polycomb or trithorax group complex, not part of these core complexes which are already shown, but they could be additional components or additional complexes which contribute to maintenance of gene silencing or maintenance of gene activation. So forward genetic screens in fruit fly, they had been very successful. You should know that Ed Lois, the one who won Nobel Prize together with Nuslin Follard and Eric Weishaupt in 98 or so, I believe. So Ed Lois discovered polycomb phenotype, the extra sexcomb phenotype, in 1960s, I think 64 or so. His wife, Pamela Lois, also used to work in his lab. And this extra sexcomb phenotype was discovered. And since then, all the polycomb members or trithorax group members, when I say all, it means a big majority, and they were discovered through very successful forward genetic screens. And you also know that these are mostly dominant mutations, and if you have them homozygous, for example, polycomb over polycomb, it's dead. If you have trithorax over trithorax, they are dead. And we used to use FLIP, FRT, the somatic clones, mitotic clones, to study their behavior in late developmental stages. Now, what we thought that, because killing these genes on going through forward genetic screen is also very, very tedious job. It's very laborious. And if we want to have a genome-wide scan of the possible additional genes which may contribute to polycomb and trithorax group gene regulation, we thought we should go through reverse genetics approach, and we decided to do a genome-wide RNAi screen or kinome-wide RNAi screen. All the kinases, all the cell signaling proteins or potential cell signaling proteins we used, and we knocked them down one by one in high throughput assay, or we knocked down all the genes in fly, and we looked at regulation of trithorax. So we started screening for trithorax group regulators. How do you study polycomb trithorax group mediated gene regulation in flies? So PRE regulated reporters are very well characterized, from Vincenzo Perota to Jörg Muller to uh, Renato Paro. So many people have designed and they have very well characterized, shown the behavior of polycomb response elements regulating, white gene. So if PRE is regulated by trithorax, the white gene is on. If polycomb is overruling and switching off white gene, you will see no expression of mini white. What we thought, we said, okay, let's have GFP instead of um, white gene, because we wanted to go in cells to do high throughput RNAi screen. You can do it in flies as well, but it's better to do it in flies, in cells. It's much more easy, fast, and then go and validate your candidates in flies. So our idea was that, by replacing GFP, it will behave similar to how PRE regulated reporters in flies behave. If trithorax will overrule, we will see GFP expression. In the absence of trithorax group regulation, when polycomb wins, we'll see silencing. So Zain Omar, she established this reporter. She started cloning, not established. She started cloning in the final year as her senior project. So our detailed construct was, we cloned BXD PRE, then imaginal disk enhancer, post pythorax enhancers. These are the enhancers. Then UBX promoter, one of the homeotic genes, and then EGFP, or we clone luciferase. Luciferase is essential because it can be quantified as well in high throughput screens. And GFP is for visual assays. Our idea was that once we will transfect this in the presence of these enhancers and promoter, there should be GFP or luciferase expression. And when we will knock down, we will reduce or deplete trithorax components. When they will be depleted, we will see down regulation and GFP expression will be diminished. So if you pay attention here now, you can see when we have PRE EGFP construct, which is basically, this construct is now called PRE EGFP. When you have PRE EGFP alone, and we had RFP construct, actin RFP, which is constitutively active, it should not show any effect when we play with polycomb trithorax, because it is regulated by actin promoter. Now in control, you can see GFP is very nicely expressed, and so is the RFP. If in these cells, we bring in polycomb, actin, so polycomb under actin promoter, a strong promoter, and you can see now, due to overexpression of polycomb, GFP expression is going down. It means polycomb is overruling the trithorax. Then also you can see the enhancer of zest. If you have EZ, which is another polycomb group complex, GFP is going down. And this is another control in which we expressed DNA polymerase alpha, are totally irrelevant, which is not member of polycomb group or trithorax group proteins. This is showing that it's not just overexpression of any protein which is going to diminish GFP expression. So this expression is, this down regulation or diminished GFP expression is very, very specific due to overexpression of polycomb group members. It means this reporter which we have designed, or which we have modified a bit by bringing in GFP, it's behaving and it's responding to polycomb doses. If you look at the Western blot, from these cells we did Western blot. So this shows you the overexpression, these cells, polycomb overexpression, enhancer zest overexpression, and this shows you the GFP. So when you have PRE GFP alone, this one, you have enough GFP. When you have DNA polymerase alpha and the PRE, you don't see much difference. But when you have polycomb and EZ overexpression, which is shown here, you see very specific downregulation of GFP. This also, what we did, we also showed that our reporter PRE GFP. So now here what we did, we used PRE luciferase reporter because we wanted quantification of our reporter. So now we are using this one, PRE luciferase. So in the presence of PRE luciferase, when we added increased concentration of plasmid, which expresses polycomb 1 nanogram, 2 nanogram, and 4 nanogram, so we see a dose-dependent effect of polycomb on expression of luciferase. So luciferase goes down as the concentration of polycomb expressing plasmid is increased. So our reporter is indeed responding to polycomb. Now, what's next? Since we were interested in trithorax group genes, we have to show that our reporter PRE luciferase indeed responds to, or the expression of luciferase, like here GFP, is due to luciferase, is due to trithorax group components. What we did, we transfected PRE luciferase in cells. Within the cell, you have PRE luciferase. And in these cells, what we did, we knocked down, let's say, we added double-stranded RNA against leg Z. In independent cells in another well, we added luciferase, double-stranded RNA, then trithorax members, trithorax, H1, Brahma, Moira, and we measured the quantity of luciferase after 72 hours of adding double-stranded RNA, 96 hours of transfection of our plasmid. So you can see at 72 hours luciferase expression, and then 96 hours. So you expect more luciferase with time, and then with specific knockdown, when you kill luciferase, you will not see any luciferase activity. And when you add trithorax group double-stranded RNA, you find knockdown of luciferase, diminished, depleted amount of luciferase. And when you compare this with a non-specific double-stranded RNA, leg Z, you don't see any effect on luciferase activity there. What we did genome-wide RNAi screen, and in a high throughput manner. We use these are 384 well plates, and each of the double-stranded RNA was spotted here in wells in triplicates. And what we did was then we added cells here, Drosophila cells. So double-stranded RNA is already present in these wells. When you add cells, these cells go within the, these double-stranded RNAs, they go within the cells and they knock down their target gene. So this way we had all the genome, double-stranded RNAs against all the genomes, all the genes in the fly genome. And after 24 hours, we then transfected our PRE luciferase plus actin renella luciferase construct. So this actin renella luciferase is just going to behave like actin RFP here. And after 5 days, we measured the activity of firefly luciferase and renella. Renella is this one. It should not show any effect because it is being regulated by actin promoter, which is not target of trithorax. Okay, and this is the summary of all the genome-wide screen. This dotted line, this one, this dotted line shows the cutoff. So all these genes we excluded using statistical assays. And what we did, we used in all these, each plate, it had leg Z, which is this one. So luciferase is very high in the presence of leg Z. And then we use trithorax and Ash1 as positive controls. Trithorax is this red and Ash1 is this one. So anything which is beyond this cutoff line, we said, which means anything which impacted luciferase activity, like trithorax or Ash1, we considered it as potential trithorax group candidate. And the experiment was done in duplicates. So you can see this is perfect experiment. We found nearly, after excluding all the there were a number of ribosomal RNA genes which mostly appear in these genome-wide RNA screens. We excluded them as well. So after filtering, we had nearly 217 genes based on their z-scores and the cutoff value. So z-score of trithorax and Ash1 was used as cutoff to say anything which affects a PRE luciferase like trithorax and Ash1, that is our potential trithorax candidate. What we did, all the genes we discovered, we tried to analyze them simply bioinformatically, and we, using string analysis, we tried to see the, based on literature search, which string works on known or published interactions. And here you can see the usual suspects. Many of known polycomb trithorax group interactors were present in our screen. There were kinases or phosphatases in our screen. Then you have RNA binding or helicases. Many transcription regulators were also present. So this gave us an idea that the candidates, all these candidates which we discovered, nearly 200 plus, they gave us confidence about this screen, that we are finding chromatin regulators or usual suspects in trithorax and polycomb group proteins. We picked up just few candidates, for example, Belchan, Babar1, Enoch, Nibde, etc. And we went back to cell lines, and once we knocked down in the cells, we compared their effect with knockdown of trithorax on endogenous targets. Here we are using not PRE luciferase anymore. PNT and PNR are basically endogenous targets of polycomb trithorax. And if you pay attention here, this is the leg Z double-stranded RNA control, no effect on PNT, PNR expression. And if you look here, the trithorax is knocking down trithorax, reduces expression of PNR, PNT. And our candidates, Belchan, Babar1, etc., they also behave like trithorax group genes. We went to then characterize a gene which is called Enoch, and Jawad Akhtar, who just finished PhD in my lab. So he characterized this gene. We went to we characterize not just Enoch, we characterize all of them one by one, but I'm going to focus on Enoch for simplicity. So we characterize all of them using extra sexcomb phenotype. I already explained you what extra sexcombs are. So in flies, for example, I told you males have this extra sexcombs. So this is the sexcomb structure present only in the fruit fly males on the first pair of leg. This is the first pair of leg. This is second and third. Second and third, they don't have they don't have sexcombs. These structures are used by males to hold female at the time of mating. When you have mutations in polycomb, you have extra sexcombs. And this is due to ectopic expression of sexcombs reduced. When I introduced Centinapedia, I told you this. When you bring in polycomb mutants, when you bring in trithorax mutation as well, these extra sexcombs, which you see in polycomb, they go back to normal. So that's why trithorax was also discovered, or they were actually discovered as suppressors in suppressor screens. And that's why trithorax was said to be antagonistic to polycomb trithorax. So we did this assay in flies. We crossed polycomb mutant alone with, so this is what we did. We crossed polycomb mutant with white 1118 mutant, and we crossed polycomb mutant with Enoch mutant. And in progeny, so from this cross, we have polycomb plus, which is going to serve as control, polycomb heterozygous, and from this cross Enoch, we are going to have polycomb over Enoch. So double mutant. If you pay attention here, so these are the, this is a severity of extra sex chrome. Minus means no extra sex chromes. Plus means, you know, this is the key. You can see. So this is severity. 4 plus means strongest extra sex chromes. We use two different alleles, Enoch 1, Enoch 2, and polycomb 1. And in another cross, we had used second allele of polycomb, PCXL5. This is experiment with PC1. This is Enoch 1, Enoch 2, PCXL5, PC. So two alleles of Enoch and two alleles of polycomb. In both, if you pay attention, so in polycomb alone, polycomb heterozygous show very strong extra sex chrome phenotype. And whenever you have Enoch alleles, Enoch 1 or Enoch 2 together with PCXL5, you can see you have this phenotype reversed to, or this one reversed to, almost none. So this behaves as trithorax group, classical trithorax group mutation. We also analyzed whether Enoch, it enhances trithorax phenotype. So this is suppression of polycomb. This is one way of proving they behave as trithorax, but they should then, we should see enhancement of trithorax phenotype as well. Normally trithorax mutant, heterozygous trithorax, it shows loss of pigmentation. So this black color is in the wild type is due to ABDB expression. Dark pigmentation is due to ABDB expression. Now, in trithorax heterozygous, you see loss of pigmentation. When you have Enoch over trithorax, you see this is enhanced. And this is the quantification. So trithorax heterozygous alone, normal flies, and then the A5. So we call this is the segment A5, A6. So when you see loss of pigmentation, which means similar to this one, this is called A5 to A4 transformation. So A5 to A4 transformation in trithorax alone is less, but when you have trithorax over Enoch, so Enoch strongly enhances the A4 to A5 transformation. And it's true for both the alleles, Enoch 1 and Enoch 2. I just told you this is due to ABDB. So here you look at expression of ABDB. So this is the, this is the wild type expression of ABDB in white 1118 mutant. You don't see any effect on ABDB. This is the ABDB expression. But in the Enoch mutant, look, you have ABDB is gone. Or in the UBX expression, which is a homeotic gene, UBX is expressed from parasegment 5 to parasegment 12. And in Enoch mutant, you see strongly depleted or diminished amount of UBX expression. Jawad also quantified expression of homeotic genes, ABDB, BXD, UBX, DFD, PNT, etc. in Enoch minus mutant, although they are dead, but he found a way by having Enoch over GFP crossed with Enoch over GFP balancer, and he collected GFP minus embryos, and he measured the in both ENOC1 and ENOC2. And here you can see in both the alleles ENOC1 as well as ENOC2, homeotic gene expression is going down, as well as non-homeotic genes. PNT is the same one which we use to validate expression our screen candidates here. So next, Jawad did immunostaining of polydin chromosomes, and on polydin chromosomes he saw ENOC perfectly and beautifully localizing with trithorax. This is these polydins, they are co-stained. So this is DAPI. DAPI is a dye which gives you, so DNA shines blue under laser 405 wavelength. ENOC antibody stain and the same polydin stain with trithorax, and this is the overlay. And this shows you that ENOC and trithorax also overlap. Then he established stable cells and Drosophila. Yeah.
सर पिछले वाली स्लाइड एक मिनट दिखाएंगे
Yeah.
सर इसके अंदर बी और सी को डी में इकट्ठे प्लॉट किया हुआ है एंटीबॉडीज इमर्ज करके डाली हुई हैं डी में
No, you same polytene, let's say this one, you throw antibody against ENOC, you throw antibody against trithorax, and finally you stain with DAPI. Now ENOC will bind wherever ENOC targets are, ENOC protein is there.
Okay, sir.
Trithorax will bind wherever trithorax is there, and then you detect ENOC with secondary antibody. So in this case, they have to be from different host. ENOC antibody is, for example, in rabbit. Trithorax is mouse, for example. So here, when you add secondary antibody, you will say secondary antibody is anti-rabbit labeled with, for example, Cy3 fluorophore. Mouse, which is trithorax antibody, you will use anti-mouse. And this is labeled with, for example, Alexa 4 or 488 fluorophore. So you will use different lasers then under microscope. So laser, when you will use wavelength 561, this will give you signal. When you will use 480 wavelength, this will give you signal. And when you will use 405 wavelength, chromosomes will look like this. This is then automatically generated. Clear?
सर ये पूछा ये ऑटोमेटिकली जनरेटेड है डी वाला
Yeah, automatically. Yeah, yeah.
ओके
Then what we did, we generated stable cells. And what Jawad did, he cloned ENOC, and ENOC was tagged with flag epitope. And he had, this construct had PMT promoter, which is a copper sulfate inducible promoter. And this cell line was selected on hygromycin. Okay. Now, after adding copper sulfate, you can see on the Western blot, ENOC is beautifully expressed 48 to 72 hours, and tubulin is used as a control to show you have proteins loaded here as well as they are equal loading in the these files. Jawad did chromatin IP, and using chromatin IP, Jawad showed that in these cells, ENOC is present in all the trithorax group targets, and one of the target where trithorax, this is an intergenic region away from bithorax complex, where trithorax is not present, you see ENOC is also absent. And what you have here is ENOC chip with anti-flag, the epitope tag, so antibody again, and then chip from endogenous, using endogenous antibody, anti-ENOC antibody against endogenous ENOC, which means not in the transgene, and also not from the stable cells, from wild type cell line. And you see very strong enrichment, and the pattern is also similar. Then what he did, he postulated that if it is behaving as trithorax, it may be counteracting polycomb. And in order to see this, because he saw already Enoch and trithorax are going together on chromatin, on polyteins, as well as in chip, what it did, he knocked down trithorax. You see protein expression of Enoch is absent. Uh, and also, since, oh, I forgot to mention, Enoch is basically already reported a histone acetyl transferase, and it acetylates H3 lysine 23 acetylation. Okay, so it was already reported by Jerry Workman's lab. So what? So this is just validation that using anti-H3K20 H3K23 acetylation antibody, you don't see acetylation anymore when you knock down. However, we knew that PNR PNT after knocking down Enoch, its expression also goes down, just like trithorax group. Now, what he showed was that if you do chromatin IP chip using anti-polycomb antibody on both PNT pipsquik non-homeotic genes and the homeotic targets of polycomb trithorax, the polycomb binding is increased or enhanced. This is Enoch knockdown cells, and you can see as compared to leg Z knockdown cells, which are these ones, there is more enrichment of polycomb on homeotic and non-homeotic targets. Now, I showed you in the beginning that polycomb trithorax also goes together with H2A ubiquitination. Okay. I showed you that H2A uh, lysine 118 ubiquitination is done by D-ring, and PRC2 complex, PRC1 complex associates with ubiquitination as well. So when you have enhanced polycomb, an obvious question he asked was if there are enhanced levels of ubiquitination, H2A 118 ubiquitination, and on these targets, you see enhanced levels of ubiquitination when you have Enoch knockdown. Now the question was, how polycomb and Enoch are interacting. On the one hand, we thought that Enoch is working together with trithorax, but what the picture is appearing, in the absence of Enoch, you have more of polycomb. Now, how Enoch and polycomb are linked molecularly? So he came up with an experiment. Before I explain that, so this shows you that when you knock down Enoch, you don't see any effect on EZ, enhancer of zest, which is core member of PRC2 complex, which catalyzes H3K27 methylation. So the level of EZ binding in chromatin IP or H3K27, there was no change when you deplete Enoch as compared to Lexi. So that led Jawad to think about that Enoch may have some polycomb specific effect. So he designed a very tricky experiment in which he did double knockdowns. So leg Z knockdown, Enoch knockdown, single knockdown, then Enoch plus EZ knockdown, and Enoch plus polycomb knockdown. And what he showed that expression of these genes, PNR and PNT, when you knock down Enoch, it goes down like trithorax. When you knock down Enoch plus EZ, double knockdown, you still don't see much difference between single knockdown and EZ, the double knockdown. Okay. However, when you have Enoch and polycomb double knockdown, you see it's just like wild type. So same is the case for another locus, which means that EZ for out of the EZ is here like PRC 2 member. This is PRC 1 member. It means Enoch is somehow having its effect on PRC1 component. And PRC1 complex, which contains polycomb, D-ring, etc., they are known to block transcription. They are known to hinder RNA polymerase 2, RNA pol 2 from gene activation. So that is what Jawad did in these double knockdown cells. So this is single knockdown, Enoch, leg Z. This is double knockdown. And Jawad used antibody against RNA pol 2, which is serine 5 phosphorylated, and antibody against RNA pol 2, which is serine 2 phosphorylated. When you have serine 2 phosphorylation, it represents an elongating RNA pol. This is transcription elongation phase. When you use serine 2 and you find polymerase to be in serine 2 phosphorylated state, it tells RNA pol 2 is on the promoter region. It's not in the elongating phase. If you pay attention here now on the transcription starts, so we use normally RNA pol 2 antibody against serine 5 to show pausing or binding of RNA pol 2 on promoter regions, and we use RNA pol 2 antibody against serine 2 to show elongating RNA pol 2. So here, if you pay attention, in the double knockdown, which is Enoch plus EZ or Enoch plus PC, so in transcription start site near the promoter region, RNA pol 2 in leg Z knockdown, there's not a lot of RNA pol 2 bound there. However, when you have Enoch double-stranded RNA at the transcription start site, a lot of messenger RNA is present, which means in wild type scenario, normally RNA pol 2 is transcribing gene. It's in promoter region, and majority of RNA pol 2, when it is gene is on, it will be in the gene body, this one. And that is what you see. A lot of RNA pol 2 in non-specific control knockdown, you see it in the gene body, not on the transcription start site of PNT. Just pay attention on PNT at the moment. Same is case for PNR. On gene body, there is a lot, on promoter region later. When you have RNA or double-stranded RNA against Enoch, there is more of RNA pol 2 stuck on RNA pol 2, more of RNA pol 2 when you have double-stranded RNA against Enoch. When Enoch is gone, positive effect for trithorax is gone, and RNA pol 2 is stuck at the transcription start site. Little in the gene body. Compare this one with this one. If you have Enoch plus EZ, Again, due to loss of Enoch, you have more in the promoter region as compared to gene body. However, when you have Enoch and polycomb both gone, now RNA pol 2 is in the gene body as well. Enoch is gone, but polycomb is also gone. So this is just like the classical extra sexcomb phenotype. When you deplete both polycomb trithorax, you somehow restore the normal kind of wild type levels of RNA pol 2 and gene is on. This is what we saw here. Normal wild type levels of both PNT, PNR. So based on this, we came up with a model that H3 lies in 23 acetylation by Enoch. It somehow counteracts PRC1 complex association or effect, because normally PRC1 blocks has an inhibitory effect, a negative effect on RNA pol 2 mediated gene activation. So when you will have Enoch is there, it's catalyzing H3 lysine 23 acetylation. It's positively regulating trithorax, and all of this is inhibiting PRC1. So this inhibitory effect of PRC1 on transcription is no more there and gene is on. However, when Enoch is gone, trithorax is there still. H3K23 acetylation is missing. Now PRC1 will have an inhibitory effect. It will be successful and genes will be off, maintained in the off state. So this is just, another slide, which is PRE, polycomb, uh, H3K23 acetylation is positively impacting trithorax, blocking polycomb group, and maintaining genes in the on state. I haven't talked about this gene. This is another gene which Heather characterized. So this is the team which did all this work. Um, so what we have seen, this is very important for you, the summary, how this whole project went. As a student of genetics and genomics, you saw establishment of a cell-based assay, which was this PRE luciferase or PRE GFP reporter. I did not share with you kinome screen, but from genome-wide RNA screen, we finally had 217 candidates. We have so far characterized 5% of them using classical genetic assay, which is the extra sexcomb assay, and they behave as trithorax group genes. I shared with you Enoch data, which have strong suppression of extra sexcomb as well as it impact the trithorax group targets, homeotic as well as non-homeotic targets in embryos as well as in cells. And finally, I showed you that Enoch, it co-localized with trithorax at chromatin, and it specifically counteracts PRC1, PRC1 complex in flies. So this is what the team. So Zen established all the reporter assay. Saima was a postdoc in the lab who was supervising Zen as a senior project, and she was the one who went to Michael Buttress, who is leading, I believe, DKFZ these days. He's one of the pioneers in double-stranded RNA screens in flies. So he helped us establishing or setting up this high throughput screen. So the screen was done there, and all the data we brought back, and we started doing work here. Dr. Aziz Mithani, he analyzed the bioinformatically, he validated the authenticity of our candidates based on Z-scores. And Jawad is the Enoch guy who picked up Enoch. He picked up Belchan. Yeah, I don't see Najma is working on another one. Zan, where's Zan? Zan is still working on another candidate. So Ahmad is there, another PhD. All these BS students, Lena worked on a candidate, Galia. So all these are senior projects, BS students who, this is MS student. MS student. Maria was a postdoc. So all of this lab, which is the epigenetics lab at LUMS, we are pretty much working on this question. And our lab was this particular project of genome RNA screen. It was not possible without funding from HEC, as well as very generous support throughout my stay here at LUMS by the School of Science and Engineering and LUMS team. So questions. Are there questions? So if there are no more questions, are you there or you have gone already?
इधर ही है सर.
जी, कोई questions नहीं है. So this was the last lecture from my side. From next week on, Dr. Afzal will start. And when is your exam?
14.
Huh?
14th.
14. So you should read पेपर्स. वो जो पेपर्स आपको भिजवाए गए थे वो आपको इसलिए भिजवाए गए थे कि आपको ये, जैसे मैंने अभी एक किस्म का ये पेपर ही सारा डिस्क्राइब किया. नजमा, शेयर विद देम द इनॉक पेपर एज वेल, जवाद वाला जो है.
Right, sir.
Send them that paper as well so that they can read this as well, because your evaluation, your exam is not about your telling us some facts. Your exam is about applying your mind and solving puzzles of genetics and genomics. Okay?
Yes, sir.
Okay. Thank you very much then. Wish you all a good day there. Allah Hafiz.
Thank you. Allah Hafiz.
Allah Hafiz.